Welcome to Medifa usmle firstaid lectures microbiology. Today the topic is bacterial structures. See, this image is showing you the generalized bacterial structure. Now we are going to study the bacterial structures in three parts: appendages, which includes flagellum and fimbriae, specialized structure that is spore, then about cell envelope, which includes capsule or slime layer, cell wall, and cytoplasmic membrane. Coming to the appendages. Flagellum. Flagellum is a locomotory organ for bacteria, so the function is motility, composed of proteins. And flagellum is very thin structure, so it is not visualized under light microscope, so it is seen under dark field microscope. Then the second appendage, that is pili or fimbriae. It is made up of glycoproteins. The function of this pili or fimbriae is to mediate adherence of the bacterial cell to the host or between one bacteria to the other bacteria during conjugation. So the function is to mediate adherence of bacteria to the cell surface of the host or between one bacteria to other bacteria during conjugation. Now the second part, that is specialized structure, which is spore. You need to answer two questions. When do spores form and which bacteria form spores? The answers are during nutritional deprivation state. To enhance the survival period, certain gram-positive rods, certain gram-positive rods during nutritional deprivation state, they form a dormant cell inside the original cell. This dormant cell is known as endospore, and this endospore is released in the environment. The spore which is released is highly resistant to heat and chemicals and even dehydration. What does this spore consist of? It has keratin like coat, and in the core it has dipicolinic acid, which is the reason for the resistance to the heat and chemicals, and consists of peptidoglycan and DNA. So we answered two questions, that is, when do the spores are formed? During nutritional deprivation state. Who will form the spores? Gram-positive rods. Few of the gram-positive rods. So which are those rods? They are Bacillus anthracis, Bacillus ceres, and Clostridium species like Clostridium botulinum, Clostridium difficile, Clostridium perfringens, Clostridium tetani. As I have told, like they are highly resistant to heat and chemicals. Do we have any method to kill them? Yes, that is through autoclave by steaming at 121 degrees Celsius for 15 minutes, and even hydrogen peroxide and iodine based agents are also sporicidal. Now coming to the third part of the bacterial structure, that is cell envelope, which is very important. Cell envelope, what is it? What do you mean by cell envelope? It is all the material which is exterior to the bacterial cell cytoplasm. It is cell envelope. What does it contain? It from outside to inside, it is capsule or slime layer. It may be capsule or it may be slime layer, and below that we have cell wall, below that we have cytoplasmic membrane. Coming to the outermost, that is capsule or slime layer. See what is the difference between these and which bacteria forms these? Let me tell you. Many of the bacteria will secrete the sticky or viscous material that encoats the bacterial cell. If that sticky material or if that material is tightly bound to the cell and is in organized way, then it is capsule. If the material that is secreted is loosely bound and amorphous, it is slime layer or glycocalyx. Now see, the composition of capsule is P for P, it is polysaccharide, and it protects against phagocytosis. Here we need to remember one of the important point, that is all the bacteria or all the capsule of the bacteria is made up of polysaccharide except Bacillus anthracis, whose capsule is made up of poly-D-glutamic acid. So which are the bacteria which are encapsulated? You need to remember at least three, like shine, or you can chin, okay? S for Streptococcus pneumoniae, HI is Haemophilus influenza type B, and N for Neisseria meningitis. The other are Pseudomonas aeruginosa, okay? Then Escherichia coli, Salmonella, Klebsiella, Group B, Streptococci, okay? At least three. Then told about capsule made up of polysaccharide and it is antiphagocytic. The third point is the clinical importance is this capsular polysaccharide is used in vaccine preparation. Not only capsular polysaccharide, along with capsular polysaccharide, we add protein conjugate because this protein conjugate will enhance the immunogenicity, okay, of the vaccine by promoting T cell activation and subsequent class switching. If you prepare the vaccine only with the capsular polysaccharide, then it cannot be present into the T cells. So what are the points you need to remember about capsule? Composition, chemical composition, it is polysaccharide. Exception is very important, that is bacillus anthracis, that is poly-D-glutamic acid. The function is antiphagocytic, and the other clinical importance is it is capsular polysaccharide along with protein conjugate are used in the vaccine preparation. And you need to remember even which are the capsulated bacteria also. Coming to the slime layer, I told the composition is same, just the arrangement, that is loose network of polysaccharide. This mediates adherence to the surfaces, and slime layer or glycocalyx, it plays an important role in the biofilm formation like indwelling catheters. We need to remember certain bacteria of medical importance like Staphylococcus epidermidis, that is catheter and prosthetic device infections, Viridian's group, dental plaques, infective endocarditis, Pseudomonas aeruginosa. You need to remember three things, that is one for cystic fibrosis, respiratory infections in cystic fibrosis, and ventilatory associated pneumonia, then keratitis and contact lens users, right? Ventilatory associated pneumonia, then in cystic fibrosis patients, that is respiratory infections, then in contact lens users, keratitis. The other one is non-typable H influenza, where otitis media. Okay. Now coming to the one of the important part of the cell envelope, that is cell wall. Cell wall is made up of peptidoglycan. See, split this word, peptido and glycan. Glycan means sugar, which forms the backbone. Okay. Then peptido means made up of peptides. So the chemical composition of the cell wall, that is peptidoglycan, consists of sugar backbone with peptide side chains. Which is the sugar backbone? That is NAM tag with peptides. What is the function of the cell wall? It gives rigid support, protects against osmotic damage. We need to know in detail about gram-positive bacterial cell wall and gram-negative bacterial cell wall. Gram-positive bacterial cell wall is very simple. It just have thick peptidoglycan layer. Can you see the image? This image is from the Lippincott. Okay, it is very clear. See, can you see the cell wall? Okay, the cell wall you have thick peptidoglycan layer. Below that we have cytoplasmic membrane. And this image is from usmle firstaid. See, this is cytoplasmic membrane and this is peptidoglycan layer, which is very thick. And you can notice this lipoteichoic acid, which is present only in the gram-positive bacteria. So gram-positive bacteria, the cell wall is very simple, only single line, it has thick peptidoglycan layer. Whereas gram-negative bacteria cell wall is little complex. Let us study that. See this image. So it has outer membrane, inner membrane. Okay, the inner membrane is cytoplasmic membrane. The outer membrane, okay, outer membrane has three parts, that is outer leaflet, which contains lipopolysaccharides or lipooligosaccharide, inner leaflet which has phospholipids with embedded proteins, that is porins, and outer membrane proteins. So the gram-negative bacterial cell wall has outer membrane, okay, which has outer leaflet, okay, with lipopolysaccharide or lipooligosaccharide, and inner leaflet with phospholipids with embedded porins or outer membrane proteins. And this inner membrane is nothing but cytoplasmic membrane. Between these two we have a space called periplasmic space, okay, and here you have a peptidoglycan layer which is very thin peptidoglycan layer. Okay, I repeat, what are the points of gram-negative bacterial cell wall? It has outer membrane, inner membrane. Inner membrane is nothing but cytoplasmic membrane. Outer membrane, outer leaflet has lipopolysaccharides or lipooligosaccharide. Inner leaflet is phospholipids and embedded proteins, that is porins and outer membrane proteins. Let us study that about lipopolysaccharides, okay, which is in the outer leaflet. This lipopolysaccharide, lipid component and polysaccharide. This lipid component, that is lipid portion, lipid A, is an endotoxin. Then polysaccharide portion is antigenic. Okay, so we are completed with the important part, that is cell wall. Now below that what do we have? Cytoplasmic membrane. We already know it has phospholipid bilayer, okay, with embedded proteins. What kind of proteins? It has penicillin binding proteins and other enzymes. Okay, we are done with the bacterial cell wall, bacterial cell structures, that is appendages, then a specialized structure, that is spore, then the cell envelope. From outside to inside we dealt with slime layer or capsule, then cell wall, the difference between gram positive and gram negative cell wall, then the third is cytoplasmic membrane. Here I want to correlate the cell wall synthesis with pharmacology. For this we need to know cell wall synthesis. We know cell wall, how it is made up of. It is peptidoglycan, right? Peptidoglycan. I told you glycan is a sugar component, that is NAG, right? And with peptides. So these are cross-linked, okay? NAG with peptides, and they are cross-linked like this. NAM and NAM are cross-linked. This is bacterial cell wall. Synthesis of bacterial cell wall occurs in 5 steps. And we need to remember 5 drugs. Which are the 5 steps? First step, that is pehla step, the enzyme is enol pyruvate transferase. The UDP NAG is converted to UDP NAM. Okay, and pehla step, that is EPT. Okay, first let us learn the enzymes. First step, pehla, that is enol pyruvate transferase. Second step, that is UDP NAM is converted to UDP NAM with peptides. So, second step is pentapeptide enzyme. Third is UDP NAM with peptide is combined with NAG NAM with peptide. This is a basic unit. It is by the enzyme bactoprenol. See, B looks like 3, right? So, the third step is by bactoprenol. The fourth, these NAM NAG, which are basic units, they are elongated by the addition of few other NAM NAG units. So this is chain elongation. It is done by transglycolase, and cross linking is done by transpeptidase. So these are the 5 enzymes. First enzyme is EPT, second is pentapeptide, third is bactoprenol, fourth is transglycolase, fifth is transpeptidase. So which are the drugs that inhibit? So number 1, pehla, that is first, that is phosphomycin. Then second, pentapeptide is inhibited by cyclosporin. Second, you remember cycle has 2 wheels. Okay. Then third, bactoprenol, B for B, B looks like 3, right? This is third step. Back to prenol is B for B is inhibited by bacitracin. Then transglycolase is inhibited by vancomycin. Okay, vancomycin. Then transpeptidase is by beta lactams. So this is about cell wall synthesis and inhibitors.